All right, so good afternoon, everyone. So just one small housekeeping note before we continue on with our lecture here, which is again just to remind you about the term quiz taking place Monday, Tuesday. So just I keep reminding you so that hopefully you won't forget to log in and I don't get emails about people saying, oh, I forgot about it and didn't do it. So log in sometime Monday, Tuesday, complete that, 10 multiple choice questions, 60 minutes. Grades will be released Wednesday, and then we shift into the rebuttal. I'll post the instructions about that rebuttal sometime, well, when I release the grades. Other than that, don't know if people are kind of starting to push along a little bit with their short communications assignments. I've started having a few conversations with people about research ideas, so that's good. Certainly you can reach out if you have any questions, want to float some ideas by me, or just talk about the project in general. Happy to do that. Are there any questions about things before we start the lecture here this afternoon? Everybody's good. Okay. All right. So the goal today is to hopefully finish up this lecture on immunity, so that way we can kind of have a nice clean break over the weekend and come back Tuesday. I've posted the notes for our next lecture topic. We spend much of the rest of the course with the nervous system, partly because there are upper year courses like D-level courses that really require that students have that nervous system knowledge. And so anytime I tried to pull a little bit of nervous physiology out to balance the course, some colleague of mine would be like, hey, students don't know this stuff anymore. And so it had to creep its way back in. So we will spend quite a bit of time on the nervous system, but hopefully still have enough time to talk about reproduction at the end as kind of another topic. So just to give you a heads up there. We covered this slide, of course, yesterday. So just to kind of get us back in the swing here, we were talking about cytotoxic T cells, and we were talking about how these cells use perforin to permeabilize the cell membrane of the target cells that they are attacking. This allows, well, not only does it disrupt the cell membrane, and that in and of itself would in part contribute to the death of the target cell, but it also creates pores through which these granzymes, these proteases, can enter the target cell and chew apart its proteins, and that will really finish that cell off. We ended just sort of asking the question, how do the cytotoxic T cells and natural killer cells that are using this perforin granzyme system to kill their targets, how do they not become victims of their own attack? And what we basically concluded here was that the cytotoxic T cells and natural killer cells are actually quite resistant to the effects of perforin. And it turns out this might have something to do with how they make this kind of rapid change to their phospholipid composition of their cell membrane. Because we saw here that when you have a cell membrane that is rich with phosphatidylcholine, that allows the perforins to nicely organize into these pores. But when you have a plasma membrane rich with phosphatidylserine instead, the perforin can't get itself organized to form these pores. And so we said that what happens with these cytotoxic T cells and natural killer cells is that as they are releasing the granzymes and the perforins, they make this quick switch to their membrane phospholipid composition, enriching it with phosphatidylserine, which then means that any perforin that does get itself into the membrane of those cytotoxic T cells or natural killer cells is not able to organize itself into a pore, and therefore the membrane of the cytotoxic T cell or natural killer cell is not going to be permeabilized. It's not going to be impacted and killed by its own weaponry. But we further noted right at the very end that this has to be a very transient change, because when you have especially a lot of phosphatidylserine in the outer leaflet of your cell membrane, that actually serves as a death signal. It actually alerts certain immune cells to come and kill that cell off. So this kind of quick switch over to phosphatidylserine that happens as you're releasing the perforins and granzymes, it can't persist. You have to switch it over while you're releasing, and then once you disengage your target cell and you're going somewhere else, you got to switch back to be a non-phosphatidylserine containing membrane. Otherwise, you're going to arouse the suspicion of other immune cells who are going to come and try to kill you. Okay, so that just kind of gets us back in the swing of where we were yesterday, and we move on now to talk about the other major type of T cell, which is the helper T cell, the so-called CD4 plus T cells. We've talked a little bit about helper T cells. We have a general sense of their role, but what I want to introduce you to here is how there are actually multiple different subsets of helper T cells that can form within the human body. And the first question, obviously, to think about is, well, when we transform one of these naive helper T cells, so never been exposed to its antigen, once it has been exposed to its antigen and it has become activated, the obvious question is, well, how does it determine which particular subtype it's going to become? And on its face, this sounds like a very similar question to when we were talking about B cells and all these different antibody types they can make. How does a given B cell know what type of antibody it should make? What class of antibody should it make? It was based on the cytokines that the helper T cells were secreting, right? And they were binding with receptors on the B cell surface. Well, here it's the same thing. We know that the helper T cells are being activated by resident macrophages and other antigen-presenting cells. And as it says here, those cells, right, the macrophages and antigen-presenting cells, they release various different kinds of cytokines, and that's what tells the helper T cell that is interfacing with what kind of helper T cell should it become, right? So again, very reminiscent of the mechanism we saw earlier with helper T cell, B cell interactions. As you can see here, there are 5 T cell, helper T cell subsets, and I just want to go through and give you a bit of a sense of the role that each one plays. I'm going to start here at the top where we have Th1s and Th2s. So these were the first 2 subtypes that were identified, and that's how they got this name, 1 and 2. You can see here that the Th1s, so it says that they are really responsible for activating the cell-mediated immunity branch of the adaptive immune system. So basically the helper Th1 cells, they are effectively activating the cytotoxic T cells. They're turning on the cell-mediated immune system. And if you jump down to the bottom of that box, they talk here about host defense against viruses and bacteria. There's a relationship here because bacteria and viruses, these are the major pathogens that are inside our cells, right? These are the major intracellular pathogens. Viruses, bacteria, they make their way often into our cells. Viruses especially, because they can't do anything unless they get into the cells and access that cell machinery, as we've acknowledged before. So when we say that Th1 cells are turning on cell-mediated immunity, they're basically helping the body to get rid of intracellular pathogens, right? Pathogens that are hiding out inside cells. We're going to turn on the cytotoxic T cells. They're going to go interact with all the various cells of our body, looking to see if any of them are harboring antigens from pathogens that are inside them. They're going to identify those that are, and they're going to kill them by the mechanism we discussed yesterday. So again, you could, if you wanted to, make a little note here. They say viruses and bacteria, but you could just add a little note here, like any intracellular pathogen, right? The Th1 cells are going to kind of help us get rid of those intracellular pathogens because they're turning on the cell-mediated immunity. They also have a bit of a role in turning on the inflammation response. We're not going to dwell upon that too much here because there are other helper T cells that are more important in that regard. But we'll move on then to the Th2 cells here. And not surprisingly, their job is mostly to activate the other side of the adaptive immune system. They're turning on the B cell immunity or the humoral immunity. And so humoral immunity, again, is all about finding extracellular pathogens, right, not inside the cell. And so that explains, too, when they talk at the bottom here about host defense against parasites, parasites never go inside our cells, right? I mean, parasites, again, we're talking mostly about parasitic worms, right, these worm-like animals. they're too big to fit inside the cells. So they are generally extracellular pathogens, and so that's why they highlight parasites here, because these Th2 cells are turning on our humoral immunity that's going to defend us against extracellular pathogens, and parasites are a major extracellular pathogen. But again, you could just make a little note beneath to say, you know, extracellular pathogens, right? Because again, some bacteria might be hiding out in the extracellular fluid, and they would also be then kind of eliminated by this Th2 activation of humoral immunity, okay? But they're highlighting parasites here because they're a major extracellular pathogen. They also highlight for us here that Th2 cells have a role to play in promoting allergy, not intentionally per se, but it's because, of course, when we talked about allergies earlier, we said it's these IgE antibodies that are kind of playing a critical role in the allergy response. Well, where do those IgE antibodies come from? They come from the B cells of our humoral immune system. So Th2 cells have that relationship with allergy because of their promotion of IgE antibody production by B cells. Now, once you get past the Th1 and Th2s, you know, just to kind of drive you crazy, there's no Th3. We go to Th17. And the reason that we have this jump here is partly historical. Th17 cells were discovered, you know, well after the Th1s and 2s. And so they were renamed, or not renamed, but named Th17, partly because they produce this particular cytokine here, interleukin 17. So that's where the 17 comes from if you're trying to rationalize the fact that scientists can't count. So what do these Th17 helper T cells do? Well, here they are actually major activators of the inflammation response, right? So they're going to start activating things like, you know, resident macrophages, neutrophils, monocytes that become macrophages, all these kind of cells that we know play a role in the inflammation response. Th17 cells are predominantly helping to activate those particular cell types. So they're big promoters of inflammation. It also mentions wound healing here, mostly because, as we have discussed, kind of the purpose of inflammation to some extent, aside from just eradicating pathogens and, you know, cleaning up any injury that's been caused to the tissues, we know that the inflammation response is basically setting the stage also for wounds to heal. And so that's where the wound healing comes from here. And then, of course, it also mentions host defense against fungi. And the reason they highlight this specifically is fungi are kind of interesting and unique pathogens in that they're sometimes some of the largest pathogens we have to deal with, right? We were talking a little bit about how fungi grow way back in the very beginning of this lecture. You know, they kind of grow through these thread-like hyphae that can kind of penetrate all throughout the place. And so because of that, the surest way, really, the most effective way of trying to kill a fungal pathogen is actually to use those extracellular traps that we were talking about earlier in the course. You kind of try to trap the fungi up in these extracellular traps, and then once you've got it tangled up, you can kind of try to degrade it with various enzymes. And so because those traps come from, you know, those macrophages and neutrophils, those cells that contribute to inflammation, that's the relationship here, right? The Th17s are promoting these inflammatory cells to become active, and then those inflammatory cells can release the extracellular traps and trap up and ensnare these fungal pathogens, and then begin to degrade them subsequently. Next on the list here we have the T regulatory cells, the Treg cells. I think we mentioned this a little bit the other day, but they really tend to be more about immune kind of regulation and suppression. So these are cells that are trying to kind of quiet down the immune system, if you will, kind of reduce its activity. And so the importance of T regulatory cells is they're important in promoting things like immune tolerance, which we're going to talk about in just a moment. You know, they're important for trying to minimize the chances of things like allergies and autoimmunity and all these things that we get when the immune system is too hyperactive. Okay, so they're kind of more of a suppression, immune suppression kind of helper T cell. And then lastly, you have what are called the follicular helper T cells, so TFH cells, which as you can see here are mostly involved in the humoral branch of the immune system, promoting the maturation and differentiation of B cells, and so kind of helping out with the Th2 cells, if you will. So those are the different classes, and again, kind of a good overview, or a good and brief overview of the general roles that they play. Again, I guess I should emphasize, like, these helper T cells, again, are not really themselves doing any of the, killing and controlling of the pathogens. They are activating the cells that are going to do that work. I also wanted to draw your attention here too. Again, I mentioned this the other day, I think, but I'll highlight it again here. When we were talking back about, M1 and M2 macrophages, right, the killing versus the repair types, and we talked about the important role that IL-4 and IL-13 play in promoting the M2, the repair type macrophages. We said again that it's the IL-4 and IL-13 that kind of promote that M2 differentiation, and said that they kind of get produced, somewhere down the line in the immune response. Here again, I can flag these two interleukins here, these two cytokines, being pumped out by these helper T cells number 2, Th2 cells. And so, yeah, by the time you've gotten to this point, right, where you're creating Th2 cells and they're pumping out interleukins 4 and 13, you're probably at the point where you want to start turning off the inflammation response and switching over to a tissue repair mode. Yeah, so next I want to talk a little bit here about really the concept of immune tolerance. And it's under the heading of T lymphocytes still here, but in reality, you could apply the same concept here to B cells as well. So what you have here in this picture in blue is you have some naive cytotoxic T cells. So they've never been exposed to their antigen before. And then you can see that they present or get exposed to their antigen here. And as we saw the other day with the B cells, when those naive T cells are exposed to their antigen, they'll undergo expansion, right? Which means they'll begin to undergo cell division, so that we now have more copies of these T cells that recognize this antigen that has activated them. And of course, this is going to help us to eradicate infected and cancerous cells throughout the body. And then at some point, some of these cells begin to die, via apoptosis. And this is a way of trying to, again, dial down the activity of the immune system. Like, hopefully we've done a good enough job of controlling the threat so it's not going to kill us, and we don't want all this collateral damage, so let's start dialing down the activity of the immune system here by killing off some of these activated T cells. And then as I show here, if you slide to the right, we might have some of these cells develop into memory T cells. And again, they're showing like they did the other day. We started off here with 2 naive T cells, and now we have 4 memory T cells for that same antigen. And this, of course, means that the next time we see that antigen again, we'll be able to launch a faster and bigger response to it. But they're also noting on this slide how sometimes the end result of this antigenic stimulation is actually that we end up with fewer cells. Here in this tolerance, you can see a lot of cells dying, and we're being left with little to no cells here that recognize this antigen. And this is the basis of immune tolerance. So memory is this idea that we are exposed to an antigen once. We are left behind with more cells that recognize that antigen after that exposure. We can therefore respond to that same antigen again, faster and stronger the second time. Tolerance is the opposite. Tolerance is that we have been exposed to an antigen, but it leaves us with fewer cells that recognize that antigen going forward. And so the next time we're exposed to that antigen, we're actually less likely to respond to it, and the magnitude of the response will be less. So the question obviously becomes, well, why would we ever want to have immune tolerance? And I'll give you 3 examples of the kinds of things or the kinds of instances where we actually want our immune system not to become better at responding to the antigen, but actually become worse and give us this immune tolerance. The first is when it comes to self-antigens, right? So our own body proteins. If our immune system, responds to one of our own proteins, we actually kind of want the end result of that to be immune tolerance, to be a reduced capacity and likelihood to respond to that self-antigen again. Because if we experience the opposite, an immune memory response, then there's a good chance we're going to respond to that self-antigen again in the future and start attacking our own healthy body cells. And this, of course, becomes the basis of things like autoimmune disorders. A second thing is food and other innocuous stuff. What I mean by that, I'll give you a little story here. So I remember when I was an undergrad, I was working in a plant research lab, and we went to a conference, which I actually think was held here, but I can't really 100% remember. In any case, there was this speaker. It was a boring conference, except for this one speaker that I remember, because he got up to give his presentation, and he started the presentation by saying, I think I misunderstood what this conference is about, because my presentation seems totally different than everything I've heard so far. And yeah, he was at the wrong conference. He was at this plant conference, but he was actually presenting on this question of how come we don't have allergic reactions to all the food we eat. And there's kind of a relationship there, I suppose. I mean, plants are food, so you can see the relationship maybe. But what I remember him talking about in that presentation is he said the dosing is really important. That because when you eat something, you're eating a lot of it, your immune system kind of thinks, oh, if there's a lot of this stuff coming in, this must have been an intentional, consumption. Let's not attack it. Let's not launch an immune response against the molecules that are in our food. And so he was trying to think about how you could use this kind of dosing idea, maybe as a treatment for allergy, maybe as a treatment for autoimmunity. So it was a really cool talk that he had given. But anyways, it kind of makes sense here that, again, if we are eating things, we don't really want, we want immune tolerance to occur. We don't want our immune system reacting to the things that are in the food we eat, just like we don't want it reacting to other innocuous stuff. I've used the examples of tree pollen and pet dander and whatever. We don't want and need the immune system to be overzealous and react to these things because they're not harming us. They're not pathogens, right? Tree pollen has never killed anybody, right? So we want tolerance in that case. And then lastly, the commensal microbes, right? All these helpful microbes that we talked about earlier that are colonizing our skin and our gut and helping to regulate our immune system and keep us free of pathogens. We don't want to be launching immune responses against them when they face our immune system. So we want our immune system to become tolerant to those commensal microbes. So this is the benefit of tolerance, right? These are the circumstances where we want our immune system not to develop memory, but to develop tolerance. What, of course, is interesting is it seems like our tolerance is decreasing. And I mean that in an immune tolerance way. Obviously, our tolerance for all kinds of other things is decreasing too. But in an immune context only, right, we are also seeing a decrease in our body's ability to tolerate things. And one of the pieces of evidence, and there's lots of pieces of evidence I could show you here, but one piece of evidence that kind of substantiates this claim is this study here from the UK. And so they plotted the number of prescriptions given for adrenaline auto-injectors per 100,000 people in the population. Of course, we use adrenaline auto-injectors for anaphylactic types of reactions, severe food allergies, bee sting allergies, all these sort of things. You can see here that over this 20-year period in the UK, there was a substantial rise in the prescription of these devices, right? Going from about 100 people out of 100,000 up to, as many as 800, right? I mean, that's an 8-fold increase in auto-injector prescriptions. Now, obviously, you could say, well, maybe these physicians are getting more comfortable with the devices, they're more likely to prescribe them. Like, you can go through the whole list here, but the reality is that at least some of this rise is probably a real rise in the incidence of these severe allergies. So it would seem like we are losing our capacity to tolerate food and other innocuous stuff, losing our ability to tolerate self-antigens. We see lots of autoimmune disorders on the rise. How come immune tolerance seems to be deteriorating? That's obviously a very important public health question. And I'm going to present to you here two hypotheses that kind of dominate the literature on this topic. The first of these hypotheses is called the hygiene hypothesis. It's basically the idea that if your immune system develops during your early childhood under quite hygienic conditions, so you're not exposed to a lot of filth and pathogens as you're growing up, then you become more likely to lose immune tolerance, right? And so the basic idea is that your immune system exists and wants to do something. So if you are growing up under conditions where you're not exposed to a lot of pathogens, there's nothing really for your immune system to do, your immune system doesn't just say, I can take a holiday. It says, I want to do something. So I'll just start attacking the pollen that comes in. I'll start attacking the little compounds from peanuts that come in. I'll start attacking the body's own proteins, right? That the immune system wants to do something, and if you haven't given it something to do, especially during the early developmental stages of your life, then it's going to find something to do. It's going to react to things that you'd rather be tolerant to, okay? So it's kind of like when you think about the old adage of idle hands, and that's where the problems come, right? If you had nothing to do, right, all day long, if there was okay, we're going to shut the university down, there's nothing for you to do. Think about COVID. I mean, you had nothing to do. You probably got antsy and restless and started doing all kinds of things you wouldn't have done otherwise, right? I mean, I was making homemade bagels. I can't do that now. I don't have time to do that now. So anyways, you get the point, I hope. Do we have any evidence? Because it's a sexy sounding idea, but do we have any evidence? And there are lots of studies that I could show you. We could go on a whole lecture of just the hygiene hypothesis, but here's one that I chose where they were looking at the incidence, the odds of you developing food allergies as a function of what kinds of pets you had growing up as a kid. And you can see here that if you had any pet growing up as a kid, your odds ratio, so your chances of developing food allergies, was actually significantly reduced, right? You see an odds ratio there of less than 1. So having any pet when you were growing up, on the whole, made you less likely to have allergies when you grew up. You can see if you start dividing it down by pets, so indoor dog, outdoor dog, and cat, right? Those tend to all have odds ratios kind of less than 1. Turtles, hamsters, birds, not as much, right? So it does seem like the type of pet might have an effect here. But the basic idea again here is that if you have a pet in your home is probably dirtier than if you don't, right? Anybody that's got dogs and cats at home knows basically you've got animals shitting in your house or outside your house. You're touching their feces, you're whatever. There's hair all over the place, and there's all this mess that comes with having a dog or a cat, right? And so if you have pets and they're making a mess, and you're therefore, as a child growing up in that environment and exposed to that mess all the time, your immune system has pathogens it might have to deal with, right? That come from your pets and all the dirt associated with them. So it is got something to do to deal with these pathogens and this filth, and then it doesn't have to worry about trying to find something to do and create these allergies. So again, there are tons of studies which have kind of tested this hypothesis, and there is a lot of support for it. There are also studies that don't support it, so I'm not presenting it here as everybody 100% is on board with this hypothesis, but there are lots of studies that seem to support this. One of the things that came up during the COVID-19 pandemic was this notion that as we were, running around sanitizing everything all the time, there was a concern that we might actually see a big boost in, allergies and things post-pandemic, because again, we were running around sanitizing surfaces, UV spraying, UV lighting our vegetables and all these things. So we were, again, really minimizing pathogen exposure, and it might lead to allergies forming. Another hypothesis that's been put out there is something called the epithelial barrier hypothesis. This is basically the idea that if our major epithelial barriers in the body start to break down, this might actually allow for more allergens and even commensal bacteria to come into our bodies and consequently trigger immune reactions. So here you can see there are two barriers they're highlighting in this figure. One is the skin, right? So we talked about the value of the skin, especially intact skin, as a physical barrier to keep pathogens and really even these innocuous things out of our body. And then they also highlight here the gut epithelial barrier. Again, we know there are lots of commensals in our gut. There are lots of things we might be eating in our food that are going to be in our gut. And what they're trying to show us here is that if you have an intact skin or an intact gut epithelial barrier, then the commensals that are in your gut or on your skin can't really come into the body and interact with the immune cells. If you have an intact skin and an intact skin and gut epithelial barrier, then allergens you're exposed to, these innocuous things, can't necessarily come into the body and interact with the immune cells because they can't get across the barrier. But if the barrier is compromised in some way, then ultimately that may allow for these commensal bacteria to come into the body, and suddenly the immune system detects them and responds to them. This may allow for other food elements or other innocuous allergens to suddenly be able to come into the body and interact with the immune cells. And so they start thinking here about, well, what factors are maybe damaging our skin and damaging our gut epithelial barriers? Here they talk about pollutants, right? The possibility that there are more pollutants in the environment that are coming into contact with our skin, causing micro damage to our skin epithelial barrier, letting these microbes, letting these allergens come in. They talk on the right-hand side with the gut that increased consumption of fats and processed foods, there's some relationship there to increases in allergic responses. So again, if the barriers that are meant to be keeping commensals and allergens out of our body are being compromised in some way, deteriorating in some way, then this allows those commensals and allergens to come into the body more readily, and they're more likely then to be encountering our immune cells, and then we're more likely to have an opportunity to launch a response against them. Okay, the next thing I want to talk about here is how we acquire adaptive immunity. And so the figure that I have here says that there are kind of, in total, 4 ways of acquiring adaptive immunity. We can acquire adaptive immunity in natural or artificial ways. And then within each of those categories, we can talk about active versus passive ways of acquiring that adaptive immunity. So let me just go through and talk about each of these 4 categories here to talk about what we're talking about. So let's start here with naturally acquired active immunity. So the example they give here is antigens coming into your body naturally, right? So you're here in this room and the person next to you coughs and they expose you to some kind of virus that they had. Antigen enters your body naturally, and then your body responds to that natural encounter by making antibodies and specialized TMB cells to help you fight off that antigen. So that would be an example of a natural active acquisition. Natural because you just encountered the antigen naturally in your environment, in your everyday life here. And active because your body actually had to produce a response. Right? In this case, it had to make antibodies. It had to, do division to create these specialized lymphocytes. So that's what we mean by the active here. So that's the natural active acquisition of adaptive immunity. Pathogens coming into your body and you responding through antibody and lymphocyte production. There's also natural passive immunity. This is where you have antibodies coming from the fetus, antibodies coming from the mother into either the fetus through the placenta, and we talked about IgG antibodies being able to cross the placenta, or antibodies from the mother going into her infant through her milk. And there we talked about IgA antibodies that are in the secreted milk. So in this case, the idea is that from the fetus or infant's perspective, It's acquiring these antibodies in a perfectly natural way, right? There's no human intervention per se. It's just, antibodies that mom makes going into her fetus, antibodies in the milk that she makes going into her infant, very natural, but passive in the sense that the fetus didn't have to do anything, didn't have to make anything, right? It's just passively picking up these antibodies as it sucks on a teat and as it just grows in a uterus that's connected to a placenta. So there's no active component there for the fetus. On the artificially acquired side, you have, again, active and passive. So artificial active would be vaccinations, right? Artificial because this is now a human, a medical intervention. We're, injecting you with either a dead or a weak form of the pathogen, or if you think about COVID, we inject you with a piece of messenger RNA that your body can turn into a viral protein. So that's the basis of vaccination. So again, artificial, because it's a medical intervention, but active because your body then has to, again, produce the antibodies and cells that respond to that vaccination. Okay, wonderful. And then we have artificial passive here, which is, again, a medical intervention. So it talks here about preformed antibodies being injected into your body. So again, this is artificial because it's a medical intervention, but passive because, again, your body doesn't have to make anything. We're just injecting you with preformed antibodies that we've taken from somewhere else. As an example of this, if you've ever had an encounter with a rabid animal, right, and there was a reason to suspect you might have rabies, if you weren't vaccinated for rabies, which would make sense because most of us don't get vaccinated for rabies on just some whim, if you had an encounter with a rabid animal, not only will they vaccinate you for rabies at that point, but then they will also inject rabies antibodies into you, right? Because it's very important that we can get that rabies infection under control ASAP before it fucks your brain up, right? So that would be an example of where you'd have this passive artificial immunity. We're just injecting the rabies antibodies directly into you from some other source. So those are the four kind of ways that you can acquire adaptive immunity. And obviously one of the questions that gets asked, and it really got asked a lot during the COVID pandemic, is, well, which is better? Getting your adaptive immunity naturally, so maybe being exposed to a pathogen naturally, or is active immunity better? Vaccination, for example, which is better? And so, again, this question came up a lot during the COVID pandemic, and so there was a lot of research that was done on it here that we can talk about. What you're looking at here, again, is a meta-analysis. So there were a whole bunch of studies where they basically compared vaccination, so artificial immunity, against natural immunity, so just being exposed to the pathogen through your day-to-day life. You can see that some of the studies were, clinical studies here done by the companies that were making the vaccine, so Pfizer, Moderna, Johnson & Johnson, and others were observational studies done by various research groups. And if you look at the data, what it basically tells us, if we put it all together, is actually there's a slight favoring here of natural immunity, right? So in terms of especially if you look at just the observational studies, all the observational data definitely favors natural immunity over artificial immunity. So if we had to answer this question, at least in the context of COVID, which is better, right? Natural immunity from just catching COVID in the environment versus artificial immunity from having been vaccinated against COVID-19, all the data suggests that natural immunity is better. Now the problem is that if you're a moron, you look at this data and you go, okay, well, fuck the vaccine then. I'll just go find some COVID-infected person to give me the virus because I'll develop better immunity to it in the long term. And obviously, that's a terrible approach to take. Shania here summarizes it pretty well. Explicitly, COVID-naive individuals should not seek infection to bypass vaccination because the risks of infection far exceed any vaccination risk. Right? So there were all these people who were so worked up about the COVID vaccine. Oh my God, it's going to do this and that and this to me. And yeah, there were some small risks associated, some heart inflammation stuff that people were finding, like little small risks. But those risks paled dramatically in comparison to the risk of the infection itself. So avoiding the vaccine, avoiding this artificial acquisition of adaptive immunity in favor of actually getting yourself sick with COVID is not a sensible strategy. You know, I remember interestingly when I was in grade 13 chemistry, and so I had to do this independent report, and I was doing it, there were people at the time who were very worked up about chlorinated water. You know, oh, if you drink chlorinated water, it's going to increase your risk of getting cancer. You know, we shouldn't be chlorinating the water. And I remember coming across, again, this one researcher who was like, yeah, if you spend 30 years drinking chlorinated tap water, there's like a statistically significant increased chance you're going to get certain kinds of cancers. But he's like, the alternative is you drink unchlorinated water, and there's a much better chance that you're going to die from some kind of waterborne illness. Right? So I don't know how we've gotten to the point where people are so worried about these small risks that they perceive in the healthcare they're receiving or the treatment of the water that they're drinking, that they're willing to ignore the much greater risk that comes from, you know, getting a virus or drinking unchlorinated water. Like somewhere along the lines, things have just gone a little bit haywire in terms of the way we think these things through. Okay, the last thing for us to talk about here, and let's see if we can do it in 10 minutes, is to talk about the concept of immune privilege. Immune privilege refers to the idea that there are some tissues in our body where the immune system cannot go. Okay? So it's weird because usually we think of like privilege as being like, you have the opportunity to go somewhere that nobody else gets to go. It's the opposite here. We're talking about immune privilege. We're saying there's tissues in the body where the immune system is not allowed to go. And so one of the places that has immune privilege in the body is our, well, I was going to say our testes, but that only applies to some of us, is the testes, okay? So if you have testes, those testes are immune privileged organs, which means that immune cells do not go into the testes. Well, why don't immune cells go into the testes? Well, they don't go into the testes because if they did, they would kill all the sperm. Because as it says here, mature sperm don't develop until long after our immune cells have gone through this maturation and education process, learning what is and is not supposed to be in our body. So they were never exposed to sperm. Our T cells, our B cells, they were never exposed to sperm as they were learning what is part of the body and what's not, because there were no sperm to expose them to. Sperm don't get produced until males hit puberty. So if immune cells were to go into the testes, they would see these sperm, they would react with any sperm-specific antigens, and they would kill the sperm, and you would be infertile. So we have to keep the immune cells out of the testes. How do we do that? Well, we use something called the blood-testis barrier. Basically, we're going to prevent any immune cells that are in our blood from being able to enter our testes because we have this barrier put between the blood and the testicle tissue. Now, what you're looking at here in this picture, so here you have pieces of the seminiferous tubules in the testes that make sperm. These kind of blue cells that you see here are the so-called Sertoli cells. And then you've got your cells here in the middle that are actually developing into sperm. The blood-testis barrier is simply these protein junctions that basically hold the Sertoli cells tight together so that nothing can pass in the space between them and get to the sperm that are deeper in the tissue. So the picture I have here on the right-hand side is just kind of showing you the various junctions that constitute the blood-testis barrier that are kind of holding these neighboring Sertoli cells tight together so that immune cells can't make their way in between the Sertoli cells and work their way towards again, the sperm that are deeper inside. You're probably familiar, if you've done any cell biology, with the junctions here. You've got the adherens junctions, right, comprised of this protein called cadherin, kind of just holding the neighboring Sertoli cells together. And then you've got the tight junctions, right, made of things like occludins, junctional associated molecules and claudins there, that are basically bringing the membranes, the plasma membranes of the neighboring cells as close together as possible, right, to really make sure these two cells are so tight together that nothing no immune cell could pass between them. So there's nothing more to the blood-testis barrier than that. It's just literally these adherents and tight junctions holding the membranes of adjacent Sertoli cells so close together that no immune cell can pass in the space between them and get to those sperm. There are other parts of the body that also have this immune privilege. The central nervous system is an immune privileged tissue, mostly because we can't tolerate having an immune response take place there. We can't tolerate an inflammation response, for example, in our brain, because there's no space, right? We know that one of the hallmark features of inflammation is swelling. There's no space in our brain to accommodate swelling. Our brain is inside of a hard cranium, so we need to not have an immune response take place in our central nervous system, so it's immune privileged as well. Our eyes are immune privileged so that we don't have immune responses happening in our eyes, which would significantly mess up our vision. And also the pregnant uterus has immune privilege, because what's growing inside a pregnant uterus? A fetus, Jesus. A fetus, it must be my last lecture of the week. It's a fetus, not feces, a fetus, that is going to have proteins expressed on its cells that are different from what's in mom, right? Because you've got these genetic contributions from dad. So again, we don't want immune cells going into the pregnant uterus and saying, what the fuck is this thing here, and killing off the fetus, so it has to have immune privilege as well. Now, the next thing to mention here, going back to the testes for a moment, is you might be thinking, well, what happens if, things are able to, immune cells are able to bypass or get past the barrier and get into these immune privileged sites? There is a backup plan in place. If we look at sperm in particular, it says here that sperm abundantly express on their surface this thing called the Fas ligand. And then if you look at many of the immune cells, many of the white blood cells, they have on their surface something called the Fas receptor. And so what happens when the Fas ligand binds to the Fas receptor is it basically activates the apoptotic pathway inside the cell with the Fas receptor. And so we won't go through all the details here because we don't really care about them, but all these things that are happening here, they're culminating in things like, the chromosomal DNA being degraded and, the mitochondria degrading, all these things. The idea is that if an immune cell makes its way to a sperm, the sperm can use this Fas ligand on their surface to activate this Fas receptor on the white blood cell, and then the white blood cell commits suicide as a result of that interaction. It goes through the apoptosis reaction. So this is a bit of a backup mechanism so that if immune cells make their way through, the sperm have some way to defend themselves. Of course, you're sitting there thinking to yourself, well, surely there must be some pathogens that have figured this out, how to deactivate these immune cells. And the answer is yes. One well-known example of this is the bacterium H. pylori. H. pylori is able to stimulate not only immune cells, but even some of your gut epithelial cells. It stimulates them to express both the Fas receptor and secrete the Fas ligand, and so ultimately then, in doing so, it causes those cells to then undergo apoptosis. So H. pylori, this is a very common bacterium. It's pretty much found in all of us, in our stomach especially, and in some cases, it will use this mechanism to kill off immune cells and even to kill off the gut epithelial cells, which can then sometimes lead to gut ulceration. Let me end here with this last little study, and then we'll be done with immune system here. The flip side to immune privilege is that areas that are immune privileged, and we've said there are good reasons for them to do this, to be immune privileged, the flip side is that these places become pathogen sanctuaries, especially viral sanctuaries. Basically, they become places where pathogens like viruses can hang out and basically know that, hey, there's no immune system coming here to eradicate me. I can just, chill here in this place, and then I could, if I wanted to, go back and reinfect the body. One study that showed this, so there was a gender reassignment surgery clinic in Montreal, and there were researchers who basically went to the clinic and they said, okay, if you have any HIV-infected males who are transitioning to females, so you're removing their testes, we want their testes, right? We want these HIV-infected testes from these individuals because we want to see whether or not there's HIV virus hiding in their testes. And so, long story short here, they found that there was in fact HIV DNA in the testes. So they found lots of HIV DNA in the CD4 plus cells. We already said those are the primary targets of HIV, those helper T cells, but they found lots of copies of the HIV DNA in what they say here, the left and the right orchid, right? That's the left and the right testicle. So basically, HIV is able to hide in the testes because it knows there's no immune surveillance there. And there are other viruses that do the same thing. Zika virus, Ebola virus, there's even evidence that SARS-CoV-2 did the same thing. Lots of people would get COVID, and then they would get COVID again and think, well, how the hell did I get it? I was so careful the next time. A lot of evidence was that it might have been hiding out in immune-privileged areas of the body and then re-emerging to re-infect you. So yeah, there's a flip side here to the immune privilege, which is that pathogens can exploit that and just harbor themselves in your body in these sites. Last point I'll mention here, then I'll shut up and we can all go home, is that the term orchid here, so orchid literally comes from the Greek, like it means testicle. The plant, called the orchid, is so called because when you look at its roots, it looks like it has testes. And so that's where the name orchid came for the plants, as a kind of parting thought on a Friday afternoon. Okay, so that's it for immunity. So we can come back on Tuesday and start talking about nervous physiology. Don't forget the quiz on Monday and on Tuesday. Don't forget about your short communications assignments, and enjoy your weekend.